at the end of last lesson, we talked about blood type, and the reason that we had a look at this is it's an example of monohybrid inheritance. We said poly or polygenetics. Poly means many, so that then means that it's many alleles controlling a trait. So something like weight or height. That's polygenic or polygenetic, which means that you've got lots and lots and lots of different loci or locations on different chromosomes that will all come together and contribute to how tall you are, which is why there's such a range. Equally, we said that there's discontinuous data that will fall into distinct categories, and those are monogenic. So they are controlled by one allele. Having a look at these, we went through these, the different blood types, and we then had a look at a Punnett square. So this would be the first time that you learned that A and B are codominant, and we talked about codominance in terms of plants versus like blended, where they we saw, for example, the white flower, the red flower, and them having an offspring that was pink. O is recessive to both of them. So when we went to fill out a table like this, we said that actually if the offspring received from one parent O negative, and they have the carbohydrates of like A from another parent, then they will have this genotype. The phenotype that they will display is blood type A. That's because the antigens here, these alleles are dominant compared to O. So this person will be blood type A. Over here, we said B takes precedence over O. Oh, gosh, there we go. So this person would have blood type B. This person would have blood type A. And again, you would have B there. And this is how you get people who, let's say, for example, the mother, if this had blood type A, then you could have one child from them who has blood type AB and another child who is blood type A, because they've received two alleles for the A blood type, and this child over here has received one B and one A. So having a look at this, just to remind you, said it on the last slide, and I think we said it before, the I there is for immunoglobulin. When you went to fill them out, you might have just written the O's down there, like so. This is the next one, and we started talking about this yesterday, and I did a terrible job of it, mentioning the idea of sex linkage. So what we said was you've got autosomal chromosomes. Autosomal are the sex-linked ones. So the 23rd pair, as you're fully aware, determine whether you are born genetically male or female, whereas there are 22 other pairs in humans. You'll get one from your mum, one from your dad, and these are what we call autosomal. They are homologous, and homologous means that, for example, if you've got one gene here, or like an allele, on this side for like blue eyes, over this side you might have one for brown eyes. They are in exactly the same place. So there might be an allele or a gene down here, two genes. You might get one allele, so one version here for like a long Roman nose. Down here from your mother, you might have had like short stubby button nose. Maybe that's just me. But whichever one is obviously then dominant will become part of your physical phenotype. These two are not counted as homologous. Actually, the XX are, because we've got, if you are female, you have XX and you've got two of them. So any genes that you have this side, you'll have the genes that side. You might have different alleles, but they're in the same locus or location. Whereas if you are male, you've got a Y chromosome that is smaller and you are physically lacking some DNA. And that then means what we're going to have a look at now. If, for example, someone inherits from their mother a genetic disorder and it's carried on the 23rd chromosome, if you are male, you are only receiving one copy because you will only have one X chromosome. So if that is a mutated copy, it might be a recessive disorder. But because you only receive one copy, you only need one allele for it to be expressed. Normally, as you know, something like blue eyes, you have to be homozygous recessive to express that allele. That's not the case in males, and that's why these genetic disorders that are found on the 23rd chromosome are so much more common in males. So what we've got here, sorry, you can hear that I've got such bad sinusitis, so I'm going to push through. We've got as we said, females, I said this to you guys last lesson, females will only ever produce X eggs. They will only ever have like the longer 23rd chromosome in there. You should be aware by now that when you are born, you have every single egg that will ever be made in your body is already stored in your ovaries. Those start maturing and being released when someone hits their period, basically gets to puberty, and those will be released. For males, they will continually produce sperm from their stem cells throughout their life. Some of those that will have the X chromosome, so those will be the longer ones, and some of them will have the shorter Y version. Those that are the Y will obviously come together during fertilization with an egg. You've got an X and a Y, and that will produce a male offspring. If you have a female baby, it's because you've had the X from the mum, of course, because that's all you could have had, and from the male gamete, which is the sperm cell. And the female gamete obviously is an egg. You've got the X, so that will then produce a female. You guys are well aware, because I mentioned to you that there are genetic disorders where that isn't the case. And for the people that I spoke to about this, you're aware of those. I've possibly banged on about them because I just find it incredible. So that's Klinefelter syndrome and Turner's, one of which you get through absolute abnormality. XXY, and then in one of them you just have an X. So this person would have a total of 47 chromosomes in total in their body. This one will have one less with 45. And that's where you get characteristics from both sexes, basically. Drawing a genetic cross diagram to represent inheritance of sex. So this is one that I drew out yesterday. For those of you who were there, I think a couple of people were ill, weren't you? This is the possible gametes that you could get inherited from a male. They could have X sperm or Y sperm. For a female, it's always going to be the XX egg. And this is the offspring that you will produce. That then means that there is 50% chance that you will have a male offspring, 50% chance that you will have a female. Any gene carried on the X or Y chromosome is sex-linked. And this is where it then comes into sex-linked disorders. Recessive conditions. So when we say recessive conditions, if you were a female, you would have to have RR for that gene to be expressed. So you would have to be homozygous recessive to have that disorder. If you are male, all you need is the one copy on your X chromosome because you have no other copy on your Y. So therefore, if you can imagine, for females, there are 3 versions that they could have. So if you are not a carrier of this disorder, you could be homozygous dominant, capital R, you don't have the disorder and you're not a carrier. You could be heterozygous, which means you've received one allele for the disorder, here we go, from one parent, and you've got a dominant disorder from another, a dominant allele from another parent. You are heterozygous because of that, and it's if it is a recessive disorder, it's not going to present because you've got the dominant allele that will take over, and that is the phenotype that will be expressed. So you do not have the disorder. To have the disorder, there's only a third of the, you've got 3 outcomes basically, haven't you? You'd have to be homozygous recessive to have it. For males, they only need to have one to have it. So obviously they could be passed from a mother, capital R, which means that it's fine, they've got a normal copy, but they're only going to need one for it to be expressed. Whereas females, quite often you might get a heterozygous female with a lowercase r there, but because it's recessive, it won't be expressed. So these sex-linked disorders, these 2 you should have been or heard of before and be aware of. Duchenne muscular dystrophy. There are lots of different types of muscular dystrophies. It's like a muscle wastage disorder. So, characteristics include, late development in children in terms of their walking, their moving, etc. The ones that we're going to focus on that you need to know are haemophilia and colourblindness. So haemophilia, most of you will probably or hopefully be aware from, maybe you talked history. And if you looked at Russia and Rasputin and all of that malarkey, the Alexander was the son of the Russian Romanov Empire, I suppose, and his son Alexei had haemophilia. And it's a disorder where if someone starts bleeding, their blood doesn't clot. So they've got this disorder, where there's just a lack of clotting factor, which obviously means if for any reason they, get a bruise, that bruise will continually bleed inside of them. So it's not something major being stabbed and they bleed to death. It's anything, their blood is just not gonna clot on its own. The other one, far less dangerous, and therefore far more common because people will not die if they've got it, is colour blindness. So having a look at this down here, you can see different versions if you've got an unaffected mother here, and then you can see here in like the green they've put an affected father. This could be for anything. You can see you could have one offspring and they are absolutely fine, and that's male, and they've inherited one Y from the dad and an X from the mum, and the mum's actually fine, so that could have produced, you know, anything. Then equally over here, if you have got like a daughter that's born, if it's a daughter, you're going to have that affected X from the dad. There's no, if this was your genotype, there's no way you could have a daughter that is not affected, because it's carried, the mutated copy is carried by the father on his X chromosome. So if you know you've got an affected father, so like I think I said that for those of you who were there, my friend who's got colour blindness, if he has a daughter, he will only, he has to be passing her that allele. However, because it's a female, his daughter won't be affected. She'll just be a carrier, because the mum, as far as I know, isn't a carrier. So if this was her genotype, then their daughter would be a carrier, but she wouldn't be colourblind because she's got the dominant allele to cover it up. Over here, when we have a look at over at this side, so my mate Harry, how he got his colour blindness, his dad was not affected, so he didn't have colour blindness. His mum was a carrier, but they didn't know that because she had a dominant allele that was fine. What they didn't know about was obviously the secret one. So when you then draw out the possible offspring that they could have, obviously you've got two of them will go over to females, that X, two of them goes over to possible males, because you've got 50% chance. The Ys will go over to the males, the X's from this side will go over to the females. When you have a look at their offspring, there is only, if this was your genotype between yourself and your partner, and you were having babies, there is a 25% chance that you would have a child who is affected by colour blindness, because you'd have one child that is absolutely fine. They've had a normal dominant copy from the mum and the dad from the X's, so that girl is okay. You've got one girl who is a carrier, so the X chromosome that she's been given from the mum is the one with the mutated copy, so the recessive copy. However, because she got an X from her dad, she's going to be fine and just a carrier. So she is heterozygous. So we've got homozygous dominant, heterozygous. Over here, this one, this person will be homozygous dominant. They don't have colour blindness. They've received a normal copy from mum, a normal Y copy from, well, I say normal Y copy, there is no copy is there from the dad. So they've only going on the fact that from their mum they've had a dominant allele. So they're not gonna be a carrier, and they don't have it whatsoever. For this version, this would be a boy. Again, 25% chance you are going to have a son who's colourblind, because to be male, you've had to inherit the Y from the dad. That then means that it's relying on whichever other X chromosome you receive from the mother, because this outcome had that one. This one obviously has this outcome. So this would leave you 25% chance that your son would have it. Your daughters in this case would not. If you had a dad who is also a carrier, so that would have to mean that this X gene was affected, because there's nothing on the Y, that would then produce a female, or that one, there we go, who's got it. But again, that's much more rare, because you have to have two recessive, so homozygous recessive, whereas this one, you just need one. Okay, colour blindness example, I think we've gone through it to death, but this is what we were having a look at before. You write these, like I said, the X and the Y. I started drawing these out at the end of last lesson. XX from the mum, XY here. With the Y, there is nothing. So it's blank, isn't it? Because they don't have an allele there whatsoever. So if you were having children, this is someone who you can see normal eye colour in the capital, because it's dominant. It's a recessive disorder. So the faulty one they've done in a lowercase n, and you're also taking into account whether they're male or female. This person, this is going to be our homozygous dominant. They are not affected. They're not a carrier. You have a second daughter, or there's a chance of having a second daughter who is a carrier, but they are not affected because they are heterozygous and they've got a dominant copy. You could have one son who is not affected at all. He's not a carrier. He has one copy, and that is for normal colour vision. Whereas you have a 25% chance of having a son. Rephrase that. You have a 25% chance of having a child with colour blindness because they've received the mutated allele. However, rephrase that. You've got a 25% chance of having a child with colour blindness. If the question asks, what is the percentage chance that these parents would have a colourblind son, then it's 50%. Okay, so from all the offspring that you could possibly have, there's a 25% chance. If the question asked, what is the chance that they will have a colourblind son, it's just between these two, so it's 50%. If the question asked, what is the chance that they would have a colourblind daughter, 0. Because this one is an OK copy, it's the dominant one, they've received that, there is 0 chance you're having a colourblind daughter. So we've got here an unaffected female because they're homozygous dominant, a carrier, so they are heterozygous, but they don't present because they've got a dominant like capital, an unaffected male here, and then that one, your colourblind male, sorry. So I spoke to you about percentages, that would be 25% chance of having a child with it, 50% of having a boy. Again, you can also write that as a ratio. So there is a 3-to-1 ratio of having a child without versus with it. Equally, you could break this down further to say a 2-to-1-to-1. So females who are unaffected, there are 2 because we've got that one and that one. Males who are not affected, there is 1, and males who are affected is 1. So 2-to-1-to-1. If you had a female carrier and a male with colourblindness, again, when you're working through these, and you should see, for those of you who weren't here yesterday, I've got homework that I'll give to you in a second. It asks you to go through all of these. In this question, they've said, oh, have they said? No, they didn't say. They didn't say, but let's say, for example, you were told in an exam that you were having a look at colourblindness, you had a female carrier. From that moment, you know it's a heterozygous female. You know you've got your X's and you know you're going to have one dominant and one recessive. Colourblind male, it's colourblind, therefore you know you've only got one copy and it's going to be one that is has a mutation, so it's recessive. So having a look at this, this is different. This woman wouldn't know, she'd have no reason really to know that she's got this X chromosome unless her dad, like she had her dad in her life, her dad was colourblind, then there's a 50% chance, as we saw last time, that she's inherited it. So she would be a carrier, but she would be able to see all shades of colour. This person would not. When they then have their offspring, you just take directly the versions of the sex-linked chromosomes that they could have. So one of her offspring or one of her eggs could have the dominant copy, one like section of her offspring could have the recessive. Again, for males, the sperm could either have a recessive copy or they could be Y and then they don't get a copy. When you then combine these two possible offspring, and you're making sure that just when you do these from the mum, start on that one, that will go to 2 outcomes. This one will go to 2 outcomes. And by the time you've gone through all of it, I said to you guys, I way prefer the Punnett squares. You've made sure that all of the options cross over. So in this one of the combinations could go there to produce a female. So to make this one, you had the X from the mum's egg, the X from the dad's sperm, and then you've got a female there. Equally, you could have the X from the mum and the Y from the dad, and then you've got a male. For this one, you put that in there, and that's the X from the mum and the X from the dad. Equally, and then you've got a female, you could have the X from the mum and the Y from the dad, and you've now got every combination that you could have had there. So having a look at this, you always need to put a key. So you don't just fill that in and like, right, we're done now. The question will have asked, well, what is the chance that they're going to have a child with the disorder or a son with the disorder? So make sure you always write a key. You could equally just do a line off it and write or explain what these are. So here, XX, it's a female. She's going to be a carrier. That's heterozygous and she is unaffected. So no issues with eye colour. This next one here, they've done it in a different order. Colourblind female, this lady. So she's received the mutated recessive copy from mum, mutated recessive copy from dad. That is homozygous recessive, so she is colourblind, but she's had to inherit both recessive copies of the allele. Unaffected male over here, they've received OK from mum, dad's Y. They've only got one allele. Fine, unaffected male, done. And then this one, the colourblind male. So in this case, there would be a 50% chance that this couple will have a colourblind child. There is a 25% chance that they'll have a female who's colourblind, or a 25% chance that they will have a male. This I've already mentioned. We've said autosomal is any chromosome that is not sex-linked. So in humans, and it's different for different mammals, animals, whatever, we've got 22 pairs. So 44 of our chromosomes are not sex-linked. It's only the final lot. And I think I said to you guys about the fact that we've got one pair, our 23rd pair. Platypuses have 10 sex-linked chromosomes. Okay, this we've already done. They keep mentioning it. You get it. Hopefully we're okay about this. This is something that we're going to have a look at again in revision, but it kind of is relevant now because we're going to talk about gene linkage. So this is what we talked about crossing over. So you should remember crossing over, and then we've got independent assortment. And this is a way to increase the amount of variation. So you can see here, if you remember, I keep banging on about it, you need to talk about crossing over between non-sister chromatids. So these two, that's two copies of the same chromosome. Those are sister chromatids. If that's from your dad, that's going to be his brown hair. There's no point having crossing over between these sisters because it's the exact same allele. Your mum, her chromosome copy would have doubled if that is blonde hair and blonde hair. If you have crossing over between these non-sister chromatids, then you're going to get what we called a recombinant, where in this version here, you would have had darker copy there, lighter copy here, and then you've got 4 different outcomes that could go to your potential children, your sperm and your egg. So what you can see, what they're showing you here is, as we know, genes are found on different locations on each of our chromosome, right? So they know where to find these things. And I always go back to breast cancer as being an example. They know that it's on the long arm of chromosome 7. So if you want to see if someone's more likely to get breast cancer, there's a specific mutation that they find on that location, and that's cervical cancer, a type of cervical cancer as well. So if you have a look at this, you can see that on this chromosome, A and B, their locus, or their loci, plural, their location is closer together than C. So if crossing over were to occur, it's more likely that you're going to have crossing over occurring between B and C than between A and B. So because A and B are really close, it's very, very unlikely that they will be separated. And this is where I Googled and found that it is correct. Often you will get people having the same eye colour and hair colour as one parent or whatever, or same as their grandparent, rather than a mixture. So they're not going to have like one parent's curly hair from one side and then eye colour that's similar to another side, because they're what we call linked genes. They're really, really close together. So you can see there it says genes A, B and C are all linked, and they are because they're on the same chromosome and they're on the same like section. There's a short arm and a long arm. Genes A and B are more closely linked, however, than A and C. B and C are actually closer, but you can see crossing over occurs. A and B stay together, but C is split up. So the further away they are, the more likely it is that they could split. Genes on the same autosomal are said to be linked as they stay together during independent assortment. So when you then make these recombinants, so this is not a recombinant, this is not a recombinant. If this chromosome went into my son, the bit of DNA that I have given him would have come straight from my dad. That's the exact chromosome that my dad would have given me. If my daughter, she had that possible outcome from the egg, this is obviously before mixing with like the dad's DNA, that would have come straight from my mother, no change to it. These ones in the middle, these are our recombinants, where you've got a bit of your dad's side and a bit of your mum's side swapping over. So this one and this one are recombinants. We then get independent assortment when they line up and it is totally random which chromosomes get pulled to which ends. And that's just reminding you of that there. Okay, if genes are autosomally linked, aka pairs 1 to 22, you won't get the phenotypical ratio that you expect. For dihybrid, you would usually expect 9 to 3 to 3 to 1 ratio. If dihybrid, again, remember we're looking at 2 alleles, or genes. I can't remember what I'm saying. It's been a very long week already. If 2 genes are inherited together, it's more likely that you get a monohybrid cross result. So again, this is like your eye colour and hair colour coming together. Dihybrid, if you've got different genes and they're not closely linked or not closely related. What have we got here? I'm not going to go through these. We'll go through these, if it's all right, together, rather than me just reading this out to you. yeah, you can read. No, we'll do these bits together. Let's do the question bits together. This I've already spoken to you about, but I'm going to cover it again now. Again, I'll go back to these questions, so don't worry about them. And we'll have a look at like doing the write-up together in those booklets. Epistasis. So not the same as epigenetics, which I keep banging on to you about. Epistasis. I suppose epigenetics. Epi is like above genetics, isn't it? It's changes to your DNA above or on top of that of the genetic material you've got. This is like above or controlling, like some genes being above others. So what you can see here, when one gene masks or suppresses the expression of another. So I gave you the example of a widow's peak already, right? So men going bald, baldness in men is a mutated allele. Some people have it, some people don't have it. I don't really, again, I can only think that it's controlled by multiple alleles because people go bald at different times, don't they? I know when I was at school, there was someone who was 17 when they started losing their hair, or their hair front started receding. Some people don't. So, widow's peak, this is the like cow's lick or whatever the flick that you have, is a V-shaped hair growth from the front. In humans, gene 1 controls a widow's peak, gene 2 controls baldness. If you have the gene for baldness, it doesn't matter if you have the widow's peak, you're still going to have no hair. Does that make sense? So you could have, it's also called like a weeping willow or like a cow's lick, that like swoosh on your hair the way that it grows rather than, I don't know, straight up, whatever. You get what I'm saying. If you are bald, it doesn't matter if you've got that. You're not going to see that phenotypical trait, whether or not you've got that genotype. So it's one almost taking precedence over another. Another example here is flower colour. So gene 1 codes for yellow pigment, and we looked at yellow and green when it came to those peas. If you remember, we said that yellow is the dominant one, so that's the dominant, and in the case of the pea plants, the R was recessive. Sorry, the green was recessive. And gene 2 codes for an enzyme that turns the yellow pigment orange. So gene 2 is also a dominant, and it turns that orange. Right, really interesting. If you don't have the dominant Y, it doesn't matter if you have the R allele, as the flower will be colourless anyway. Gene 1 is epistatic to gene 2, as it can mask the expression of gene 2. That masks the expression of that. That, if you've got it, it goes orange, but if you've got that, it's yellow, is what it's saying, I think. Crossing homozygous parents. So homozygous, if you remember, we're talking like our wild types, I think, where you've got capital Y, capital R versus lowercase y, lowercase r, will give the ratio of 9 to 3 to 4. So 9 of them will be orange. Then you'll have 3 of them will be yellow, and then you've got the recessive ones that will be white. Phenotypic ratio in the F2 generation. So again, I'm just going to repeat if anyone wasn't here. Your wild type parents are the ones that, to have a look at any like genetic traits and how they're inherited, whether they're recessive or they're dominant, you have to get them to start with. So they would be like totally like homozygous. So I'll just write it out rather than try and explain it. In this case, one parent that you're starting with would have dominant. Equally, you would cross that, and this is a dihybrid cross because you've got the multiple genes that you're looking at. You would cross that with a parent who has got recessive, right? I said to you guys, it's far more, it's less complex in the exam questions. It's not as hard as taking all of these into account. So, you know, please don't worry. We then saw on a video and saw some examples in this slideshow about all of the different like possible offspring that you could have received. We call that the F1 generation, so the first set of their babies that they produced. When you then cross the F1 generation, so F1 and F1, you then get what we call the F2 generation. And so often, if you remember when we talked back to the pea plants, you could have green, so green, yellow, they all have the same baby with the dominant yellow. When you then go to the F2 generation, that's when you see the 3-to-1 ratio. Okay, so this is, if you were like, I think this is a really good way to understand it rather than reading, draw it like so in the exam. So if you were taught through this information, you're saying that there's a molecule to debate whether or not there's colour. If they don't have it, they don't have it. You're not getting past that stage and you're going to have the outcome of white. If they've got gene 1, that will get them to the yellow pigment. However, if there's gene 2, if they've got like the dominant allele for that, then it's going to turn it to the orange pigment. So let's say, for example, you had something that was YY RR, okay? If we start here, they've got dominant Y, which means, yes, they're going to be able to have yellow pigment. Here, however, they've got recessive, so they're not going to get past this bit. Oh, gosh. Oh, what have I done there? There we go. So your plant there is, in that case, is going to be a yellow outcome. If you had recessive, capital, like dominant recessive, in this case, because they don't have a dominant yellow, they're not going to get past this point. So it doesn't matter that they've got the red, for the orange pigment. Does that make sense? They're not going to get there. So that would be white. If they were homozygous dominant and they had yellow in the capital, they would get to this point. If they've got capital, if it's either 1 or 2, because it's a dominant, they would then turn out orange. Does that make sense? Hopefully. Right. Work out the gametes of the alleles. Oh, again, I mean, we've already just done it, but you can see here, capital, so along these lines, it's going from colourless to the yellow. Then RR, that means that it goes all the way to the orange. This one, YYRR, it's going to be totally colourless. So our F1, these are your wild type parents, aren't they? Where you've got dominant, recessive. The offspring will all be this mix. So that's your F1 generation. When you cross those, incest, when you cross these plants, they are heterozygous for both of them. You then get these possible gametes, gametes that are going to be passed on. So you could have both of them that are dominant, one recessive, one dominant, or both recessive. That's what you could possibly get from one parent, from the gametes they produce. And then this one, exactly the same. They could give you 2 capital, 2 recessive. I always deal with 2 and 2. So as in like 2 of the same. So you've got 2 of the same, they're both dominant. 2 lowercase, they're both recessive. You could have a mix of capital Y and lowercase r, or lowercase y and capital R. Oh no! Oh, you know, I don't do computers. When you then filled out this Punnett square, this is where we get this 9-3 whatever ratio, or in this case 4 ratio, when you looked at how many could be produced. Dominant epistatic alleles. If the epistatic allele is dominant, then having one copy of it will mask the expression of the other gene. That makes sense, doesn't it? We already knew that already. You're just talking about not over an allele, so that takes precedence over this allele. We're talking about this gene, or this allele takes precedence over even these two, is my finger showing that? These 2 genes. Crossing homozygous parents will result in a 12-3-1 phenotypic ratio in the F2. Squash colour. Coloured allele, you could have this next. So first of all, does it have colour, does it not? If it has a capital, like W, then it will be white. So this is like literally like pumpkins we're talking about, right? So you know that you've got like, that's what I think it's talking about anyway, like orange type pumpkins, that kind of species. So it's dominant apparently for white. So if at any point it starts with a capital, it's going to be white. It doesn't matter about this. That one's taking precedence. So you go straight to white. This reminds me, you guys don't have it, but you used to have these flow diagrams in a magazine. Like, does he fancy me? Yes, this. Oh, tragic. But this takes me right back, right? So if you've got a capital W, it's going to be white. If it's double, like homozygous recessive, it then comes into whether or not there's a capital Y to make it yellow, or the next recessive copy to that is the green, like so. Okay. Complete the cross. Again, this is just showing you, The F1, again, these are the wild type parents where it's totally dominant, totally recessive. You always get this mix. F2 generation, you get all these possible outcomes of the capital and the capital, the lowercase and the lowercase, the capital W, lowercase y, capital Y, lowercase w, and exactly the same that side. When you put it into a table and fill it in, 12, 3, 1 is what would be produced there. Okay, no, I'm not, like I said, I'm going to go through these later on, some of these questions when we are back together. So ignore. Oh my days, loads of the problems. Oh my right, okay, let's skip past these. Here we go. Chi-squared, I'm going to come back to chi-squared with you guys. Right, this one, let's go into this one. So we are 35 minutes in. Great. The factors that can affect the evolution of a species. This should be pretty easy for you guys. You've done the the ecology topic. We've talked already before about, allopatric and sympatric, all of that malarkey. Standing order of height, let's not do that. Oh my days. Stand in order of height, stand in order of shoe size. I can see this is talking about ranges, isn't it? Okay. Oh, they really like that image, right? If it's attached or detached, that's recessive. Two people are the same. Why? And it's the idea that we have multiple alleles, or multiple genes controlling some traits. We've got multiple alleles within those genes. Oh, why does that keep happening? Okay. Nope. Continuous, discontinuous, we've already talked about that. Can you give some examples? What is selection? Hopefully you remember natural selection. This was Darwin and his finches. So Darwin, so famous, came up with evolution by natural selection. He travelled on a ship called the HMS Beagle. He went to the Galapagos Islands, and that was really cool because everything he found there was totally different to what we had in the UK. And when he went there, he found that there were these groups of birds on different islands. So they'd been isolated on different islands. And when he studied them, he found that they all had slightly different beaks. And what he found was, depending on which island they were on, they had different beaks according to the food type that they ate. So if they had short little tiny sharp beaks like this, they might be pulling out insects from inside a tree. If they had really strong, thick beaks like this, they were probably cracking open Brazil nuts or that kind of thing. And so that they had changed, accordingly. This, you should remember from GCSE, a guy called Jean-Baptiste Lamarck. Lamarck's theory was basically use it or lose it, and I feel so sad for him because he wasn't actually wrong. The only thing that they had no evidence for at the time, let's rephrase. He suggested that if an animal was using something, like a giraffe using its neck, it got longer over time and it would then pass it on to its babies. If it wasn't using anything, then it wasn't passed on. So we do kind of have evidence for that in things like our we are no longer using our appendix. Our appendix is getting smaller, and one day maybe it will disappear from the human body because it's not being used. Giraffes using their neck, etc. So what they did to prove whether or not this was right, they did things like they got mice, they chopped off a mummy mouse's tail, so she had no tail. They then got her to have babies, and what they found was, of course, the babies were born with tails. So that was proof that he wasn't exactly correct, because actually what he said was, if they were using that trait, it was passed on to their offspring. However, obviously then Darwin came in and said, this is what happens and it takes millions of years. He wasn't exactly wrong, because we now know about epigenetic changes, where you had people, and I'm sure I've gone on too many times to you about it, where you've had people for example, a famous case, in a prisoner of war camp, they had malnutrition disorders like scurvy, rickets. They then got out of the war was over, they were freed, they started families, they were back to eating a normal diet. However, these same disorders of malnutrition, like rickets, etc., was found in their grandchildren. So 2 generations later, even though their offspring, so the parents of these children and their grandchildren had, nutrient-rich diets, because those changes were made to their grandparents' DNA, it was passed down within 1 or 2 generations. So actually, he wasn't totally incorrect there. Most populations have relatively stable size. All organisms produce more offspring than can be supported by their environment. So this is really, makes me really sad. You can see there a frog. They produce hundreds of eggs, so they have multiple chances of offspring surviving. And the reason they produce so many is because so many of them will be eaten and killed and died. It's a bit like if you think about little turtles. You see these tragic videos of them hatching out, crawling from the sand to the sea, seabirds coming and eating them. So they're not going to survive. So they'll have loads of babies purely because a lot of them won't make it. Whereas with humans, we're okay having 1 or 2 children, because actually we as a species are really good at surviving. And this is what Darwin also said. He noticed that in terms of how many puppies a dog has, etc., that there will always be more offspring than will end up surviving. So there will always be the runt of the litter that will die. And he acknowledged that there is competition between members of the same species for everything. So if you remember, we've got interspecific competition, which is between different species. So say, for example, you've got a frog and it's trying to compete against, a snake to try and catch something to eat. They're different species, so it's interspecific. If you've got a frog versus a frog, they're the same species, so it's intra-specific competition. Within any population, there is a gene pool with a wide variety of alleles. And variety is the word that you keep coming back to. You have to say that mutation leads to variety because it produces different alleles. So you'll have some alleles caused by mutation, which will produce a phenotype of frogs with larger legs, or you will have some with, let's say, an allele for larger eyes. Some individuals have combinations of alleles that make them better adapted to survive. This one. Therefore, they reproduce and pass on these successful alleles to their offspring. Any that do not get alleles that are a selective advantage in their environment, they die, and those alleles are then not passed on to their offspring. Types of selection. Directional selection, individuals favoured in one direction. Stabilising selection, average individuals favoured. Can you think of any examples of these? Okay, individuals favoured in one direction. So an example of directional selection. Number of individuals up there. You can see then temperature. This is an enzyme optimum temperature graph. If it gets too hot, it denatures. Too cold, it denatures. There is a middle point, which is your optimum. Selection pressures. If you are going this side, so we can see that it's hotter, warmer, whatever. If the temperature falls, individuals with longer fur length are at an advantage as they have better insulation against the cold. Right, sorry, fur length along here. Temperature, I'm just trying to understand this graph. I don't think I know. Oh, optimum fur length at 10 degrees here. Fur length along here. Oh, that's the temperature curve. Right, optimum length is here. Right, so if it gets hotter, are we talking along that way? Hotter or colder? I'm not really sure which way we're going along there, but okay, right, bear with. Okay. If the temperature falls, the individuals with longer fur are at advantage as they are better insulated against the cold. I get that. There is a selection pressure favouring the animals with longer fur, so the animals are more likely to survive and reproduce. I get it. Over several generations, the average fur length increases as more young have inherited the gene for long fur. I also get that. When the mean fur length has reached the most advantageous length, the selection pressure ceases. Yeah, that makes sense. So it stops getting longer. They all get to a point where they've reached the like most of that upsets me. Most optimum length of fur for a rabbit, and that is like the general, like for the UK. So this is where we will have like different length fur in the rabbits in this country to another country where it's colder, for example. So that was it, all going in the same direction. So they all merged to have the same length. That was your directional change. We're now going to talk about stabilizing. In years when it is hot, short fur is favoured. In years when it's cold, long fur is favoured. So this is our bell curve of, you know, you'll have some with really short fur, some with long fur, and some in the middle. When temperature remains constant, individuals at the extreme will never be at an advantage. So if you can imagine, if you're not having extreme winters, extreme like hot summers or whatever, these guys are gonna die because it's too cold, die because it's too hot. The mean will remain the same, but there will be fewer individuals at the extreme, because as you can imagine, if it's super cold, anyone with really short fur are going to die. They won't survive the winter. Anyone with really long fur and it's like super hot, they're going to die. So you get more, it's less of a bell curve. You don't have these extremes. It's only the ones in the middle that are going to remain. And that is what they're saying is the total opposite. So one of them, directional, is where they all merge together in the same way until you reach a point. Then the stabilizing selection, they all start having the same or similar alleles because the extremities like die out. Right, 15 minutes remaining. I want to see if we can get through this. These guys, I'm sure you've heard of these before, haven't you? I'm definitely doing these with my year 11. So if you haven't, I'll just remind you. Factors that affect the evolution of a species. The founder effect. I always think about, you know, founding fathers of America is where one or two or a few from an individual like species moves to a new area. Okay, so if you think, for example, it's like humans taking rabbits and putting them in a different country abroad. Equally, you've got the bottleneck effect where they all start merging and looking similar, and then genetic drift. And I'm going to show you some examples of that in a second. So what we can see here, genetic bottleneck, you've got parent population, there's loads of variation. There becomes something, like you can see there, drastic reduction in population. There's only a few surviving individuals, and the ones that survive could then change the next generation. So this is caused by some kind of disaster, for example. So really, really sadly, the one that I always think about here is this must have happened after wars. So if you think, if you've got loads of variation in men, loads of them who are fit, healthy, well enough, fit enough, didn't have, you know, some kind of mutational disorder that meant they didn't have to join the war. Oh, you only had some left behind that couldn't. The ones that came back from war, I mean, it's not as straightforward as that, is it? But, you know, will have come back for certain like traits that they had. So maybe they were really good at running or really fast, or maybe they were really aggressive, so they attacked first rather than waiting to be attacked. Really grim example, but you see what I'm saying. Also, the ones that you had left were the people who were not fit enough or strong enough or whatever, genetically, that it wasn't okay for them to go and fight. So that is massively going to affect the next generation because you've got fewer options. There's way less. Oh gosh. There's way less variation in the type of offspring that you could have. Oh my gosh, I now don't know where that was. Okay, something like that, done. Right. Original population, large numbers of the population dies. That's some catastrophic disaster, reduces the population size. Some alleles are lost. They then reproduce with whatever is left. And then you've got genetic diversity. New population genetic diversity is greatly reduced. So you've only got those remaining. There's less of them to reproduce from. The elephant seal. I want to say that's the founder effect. Lord knows. Or was it talking about genetic drift? I mean, I don't know what would choose to make with that, but there we go. Right, whatever. Original population, a couple of them break off. So you've got these guys moving abroad or moving to a different area. You then don't have the same variation that you had to start with. And the new population, what you can hopefully see is it's created, well, eventually we're going to get to a new species, but it's very different because there's way less mutation or less variation than what you had in the original population. Over time, that could produce two totally new species. Draw your own founder effect diagrams. Hopefully you did, you get it, fine. Genetic drift said that characteristics are passed on by chance rather than due to factors that affect the individual's ability to survive and reproduce. passed on by chance. I want to come back to that. Actually, okay, is that what they're saying? This finished? Right, okay, I'm going to 49 minutes, call it there. I'm not really happy with what they've said there about genetic drift. I want to come back to you guys about that, so we will do that in the next lesson.